Hello everyone, I'm Haryana Thomas, and today we'll be talking about modeling extracellular matrix accumulation and kidney damage. I worked with Dr. Fibrosis at Oklahoma State University. So the objective of our research is to study the spatial distribution of excess extracellular matrix so that we can gain insight into the progression of kidney damage. So the motivation for our research rises from the widespread prevalence of chronic kidney disease in the U.S. 1 in 3 adults are at risk for chronic kidney disease in the U.S. and 9 out of 10 that do develop CKD are undiagnosed, making CKD the 9th leading cause of death in the U.S. And one of the major causes of chronic kidney disease is diabetes, a disease for which treatment methods and diagnosis methods are still lacking due to the complexity of the kidney and cellular processes within. So we hope to develop a computational model to elucidate these complex processes so that we can understand kidney damage better and develop better treatment and diagnosis methods. So our model is of the glomerulus, which is the filtration unit of the kidney, and here we have a healthy glomerulus and compared to a diabetic glomerulus. And one of the the hallmark of diabetic glomerulus is that it has extracellular matrix accumulation within and around the mesangial cells. And so what happens is the high glucose in the capillaries diffuses into the mesangial cells, causing dysfunction, and then the cells produce excess extracellular matrix, excess collagen, which causes extracellular matrix accumulation. And so we've used CompuCell 3D to capture the following processes. So here we have a diagram of our model where on the left side is the capillary from which glucose fluxes in, interacts with the mesangial cell, which causes the mesangial cell to produce excess collagen and cause extracellular matrix accumulation. And so here we have our model with multiple cells and no fibers initially, and this model shows the diffusion of glucose into the mesangium, and the cells respond by producing collagen. Here we can see the cells that are producing collagen, and what we see is that the cells that are closer to the capillary, which is the left side here, produce more collagen, contributing more to extracellular matrix accumulation and kidney damage. And then we incorporated the fibers into our model to see how the fibers affect the spatial distribution of the extracellular matrix that was being accumulated. And we saw that the fibers have a slight effect, but the spatial distribution is highly dependent on the spatial distribution of the collagen, the soluble collagen here that crystallizes onto the fibers. So in the future we hope to validate our model using visual data from either rat glomeruli or human glomeruli. And thank you for listening.